Yeah. Yeah.
Excuse me.
I think, yes, you can use the pocket.
Good morning, everyone. Welcome to the second day of ESSO. I'm glad to have this opportunity to introduce Dr. Federsel. Dr. Federsel got his PhD in organic chemistry at Royal Institute of Technology. And I just noticed a couple of publications with Jan Bergman, who also work with selenium, tellurium. We have several professors here who also work with selenium, tellurium chemistry. And he started as process R&D chemist in Astra. Astra is the company which later became AstraZeneca. And he dedicated decades of experience in chemical process field. So, and he became a director of science, followed by appointment as senior principal scientist. Academic qualifications lead to associate professorship and seat of the board of the School of Chemical Science Engineering. 2009, he was elected to the Royal Swedish Academy of Engineering Sciences. And also the curiosity, this Royal Swedish Academy of Engineering Sciences is one of the oldest Royal Engineering science to the Swedish is the oldest one in engineering sciences. And in early 2017, he decided to return to his home country, Sweden, again. And he also developed a biotech startup called Enzyme. And nowadays he's sharing the experience to industrial organizations and academia. I have another slide here just for curiosity. I found this article he published in 1993 about asymmetric synthesis and the importance of chirality in an industrial approach. And I guess he predicted the topic for the Nobel Prize in 2001. So, and his contribution to academy was also present in very important articles during the 90s and recently he published very important articles in high impact journals. He also published a very interesting book, Asymmetric Catalysis on Industrial Scale. So please, let's welcome Dr. Federsel. Thank you.
Thank you. Thank you. Thank you. I hope the applause will stay the same or even be more intense when I'm done with. So that's a better sign of your appreciation, because I'm here for you. I'm here and want to talk about what I'm passionate about, namely process research and development. I've devoted all my professional career to this topic, and I haven't had a day where I didn't learn something new, and I hope that will be possible for you to feel that, my enthusiasm for the topic, and maybe give you an insight into what process R&D is about. I think Alvaro had done a great research job finding out all sorts of aspects of my life. What he didn't tell you about was that my family situation, I'm happily married with the same woman for 38 years, so I'm pretty proud of that. Especially in Sweden, there are so many divorces. People just think they can find better deals anywhere else, but 38 years, I have 3 children, 2 boys and 1 girl. 2 of them are married. I have 2 grandsons. I love dogs. I have my 3rd Labrador. Boris is his name, nothing to do with the current political figures or so. I named him Boris when I got him 12 years ago, so he's my best friend. Keeps me fit as I need to exercise him. I love wines, so wherever I go into a wine country, I try to exploit what they can offer in wine. And then I'm a passionate pianist, so I've actually performed in the Stockholm Concert Hall. I made my debut there quite some years ago, so I'm a classical educated pianist as well. That's me in a nutshell. But chemistry has taken a very big part of my life, you can imagine that, and even if I might have reached the age where I could retire, I don't want to. The chemistry is so fascinating and it happens so much, and I feel I can give to chemistry, to new generations of chemists, this enthusiasm that I felt all these years. And well, you will judge. You will tell me afterwards, or tell the organizers that was really good or that was lousy. It's up to you. I'm here for you. I prepared a decent number of slides, well over 100. Keep us going. We will not run out of slides. I'm pretty sure about that. Maybe we will not cover all of them. But the good news for you today is that this slide pack will be made available to you in its entirety as a PDF file. So you don't need, I understand it's a frustration to sit and make notes, etc. And some synthetic schemes are quite busy, so it's better you focus on me, what I tell you, and you know you will be able to review the content on your own time, so to speak. We have a number of hours ahead of us. I've thought about how to do this in the best way. It should be digestible for you. You must feel comfortable seeing me here, talking to you, etc. I will give you opportunities to ask questions. We cannot keep all the questions till the end when we are all exhausted. We need to take them as they appear, but maybe not all the time. It will be a bit disruptive, which is not really what you like. So I'll try to make a pause after each discrete package, and which will not be too long, and then you can have the opportunity to ask questions which belong to that particular area. Is that a good deal? Are you on board with this? Don't be shy. Ask. I mean, I'm happy to answer, and if I can't answer, I can probably look it up somewhere. So let's stay a bit interactive. Also, I might use expressions. I'm a bit spoiled after these many years in the business. So we all have a jargon using words that you might not be familiar to. Stop me and ask, because it's the outcome of this long day depends on if you feel it's been worthwhile, if you understand. The area that I will talk about, yes, this is a synthesis workshop, but I tried to expand it. It's much more to developing a medicine than just organic synthesis. It's important, and I will have that as the core part, but I will also talk a bit about the pharmaceutical industry, a bit about safety issues, hazards, quality. Doesn't sound so sexy, not compared to synthesis, but it's something you need to be very mindful of. A bit about the regulatory constrictions. I would guess that most of you have not been exposed to, let's say, the intrinsic pharmaceutical environment, so you know the details, what's actually happening there, what are they doing, why is it so expensive, etc., etc. Why are they so unsuccessful? So this cartoon here, I love that one, because you always need to explain to your supervisors, to the management of a business, why should they invest in you? Why should they give you money to have a group and buy equipment, etc.? Well, for process R&D, which is a bit technical, for most senior executives in the pharma business, they hardly understand. Well, chemistry is a bit an outlier, but process R&D is even worse. It's too technical. They talk about factories, manufacturing. Yeah, don't bother me. They want to have the medical results. They will show it's an efficacious drug in man, etc. So these are just supporting functions. And this is a cartoon that shows, I've used it several times. I unfortunately don't know exactly who painted it, but this shows if you skip process R&D, yes, the cost goes down, but look at what happens. I don't think that anyone would like us to produce large-scale compounds using laboratory equipment, and this is what it's all about. So I would have an introduction. We have to keep a relatively fast pace to go through as many as possible of these, but I'll try to talk calmly and not too quickly. So hope you can all follow me. How do you do it when you take laboratory chemistry to manufacturing scale? Synthetic chemistry and route design, that's a core part. We're talking about how to make the molecules, the active ingredients. Sustainability, that's in every news program more or less, all around the world. Everyone talks about sustainability. What is the downside is that very few of the people who listen to these programs and who make them know what sustainability means, what it entails. What does it mean to become green from a chemistry point of view? I will talk about that. Hazard prevention. Chemistry is a dangerous science, as we know. A few facts about that and what we need to be mindful of. Flow processes. I know a few of you in the audience are involved in flow processes. This has also made major inroads in the pharma industry. Quality and regulatory, I mentioned that. Again, not maybe the most hyped areas, but extremely important. And a few conclusions. Is this a good menu? You think that will keep you alive and going? And we will have a couple of breaks. I already mentioned that. So I won't keep talking for 6 or 7 or what number of hours. I will make a few breaks. So we are on a long journey, not only today, but to develop a novel medicine takes an enormous time, and I know that Sir Simon alluded to this yesterday. I could spend the whole lecture talking about this and what's been done to shorten the timelines. I know Pfizer has had programs. Whilst I were in AstraZeneca, we had so many efforts and initiatives trying to cut. Why does it have to take so many years? That's the constant question. You can see the years in discovery. I mean, these are ballpark figures. There are outliers, etc. Some manages once in a lifetime to do it half time, etc., etc. But this indicates what we are talking about. You enter, this is the early stage when you're looking for a molecule that would suit you, as we heard elegantly about yesterday. Then you go into the development phase with the 3 clinical phases, 1, 2, and 3. The number of compounds that are on the desk goes down sharply here. One of the major enemies to the pharma industry is the attrition. Attrition is a great English word that I feel really fits this purpose. It's the loss. It's the opposite to success. So we lose most of the projects that we initiate up in this phase here. Most of them are gone. That doesn't mean we cannot learn a lot. We do learn a lot from every project, even if it doesn't enter a clinical stage. Chemistry is not the main blocker. Chemistry is an enabler. Chemistry can allow other people involved along this timeline to really achieve something, to test hypotheses, as we heard about yesterday. So it was a very good introduction to what I do. At the end, there will be an approved drug, hopefully. Over all the years that I've been involved with drug development, we have constantly discussed, who is my customer? Who am I doing this for? Why am I doing this? Besides having a good time working with interesting chemistry, interesting molecules, why am I doing this? It sounds as a very trivial question, but it was often ignored until, at least in my company, previous company, a few years ago. We do it for the patient. The patient is the ultimate customer, and I guess you agree, Simon. Yes, it's the patient. But the patient does never appear in any development program only as a victim, in a sense, as someone that you use to test your medicines on. But I've seen a few video recordings when I still was in AstraZeneca, where they actually interviewed patients who had taken the medicine, and they spoke into the camera and gave feedback how, in our case, the AstraZeneca medicine had affected their lives. What had changed? Not always positive, but many times positive. It improved my life. I have new hopes for my future. Cancer patients on a death row, more or less, who managed to survive and have good quality of life. That is a very strong message. And it convinced many people who were a bit skeptic. It's just to get the paycheck, but you really contribute to people's well-being and survival. I think that is a very important message. So given that context, this picture that we are still looking at, in spite of many efforts trying to improve and smoothen out and cut the corners and what have you, at the end we get out the medicine that will, in the best of worlds, life-saving. Why do our projects fail? This is published data a few years back, but the picture is identical today, I would say. And Simon actually underscored that the main hurdles is the efficacy in man and safety. If you sum these figures up, it's well over 70%, maybe 80%. Those are the factors that kill projects, not the chemists. I have never in all my years been involved in any project where the chemist said, we cannot make this molecule. We always deliver. We always deliver. Sometimes with an enormous effort, but we do. The problem is that predicting the efficacy and avoiding safety issues, you would like to do that so you don't have to go through all these long testing periods. But our understanding of biology, toxicology isn't developed enough. So we cannot make any projections. So you have to do it the hard way. So when, if that will ever happen, we can make better projections on how a particular compound will work in man. A lot is saved. You have a few other reasons as well, but efficacy and safety is what you should remember from this. Here are a couple of molecules. I've been involved in many of them. AstraZeneca success stories. Why I'm showing this? Not to brag about it per se, it's more the diversity. If you look at these structures. Small molecules, I'm a small molecule man. Doesn't mean I'm a small chemist, but I'm a small molecule man. The diversity. We spoke over the pizza yesterday about finding compounds with some sort of pharmacological effect is not the hurdle, it's not the major, it's to find the right compound with the right properties, and I will in a slide or two show you what are these properties, what are we looking for. I will talk more about omeprazole. That's a flagship in our portfolio for good reasons, and it's one of the best processes we ever developed. This molecule you saw yesterday, a calcium antagonist belonging to the amlodipine family in a sense, not as successful as the Pfizer product, but still doing pretty well. This is a local anesthetic. When Astra is a very old company, formed more than 100 years ago, but it started to really grow when we launched the world's best hitherto local anesthetic. We are a world leader. We were a world leader in local anesthetics. Xylocaine, for example, launched in the late 1940s actually. That's started the upwards trending curve. This is late coming analog to xylocaine. The reason we have homed in on one of the enantiomers is that the opposite enantiomer has a heart toxicity. These are single dose compounds, so, but still, people who undergo surgery, you don't want to expose them to any unnecessary side effects. And with this heart effect, it was thought it's better to home in on only the local anesthetic part and skip the wrong enantiomer. Penicillins, I will come back to this very fond molecule of mine that I spent quite some time working on. Metoprolol, this is a medicine that I have to swallow every day now myself. It's a beta blocker. It's against my heart arrhythmia, something that many people of my age group suffer from. So it's good. It's nothing spectacular from a taste point of view. I've had worse experiences, but it really works. It's a beta blocker, and it's been very successful This is a steroid to treat respiratory conditions, this as well, and this as well. So, if I wouldn't have told you, you wouldn't. If you had seen a cohort of molecules, maybe one or two of those look suspiciously similar to what you could imagine to be a drug, but otherwise you couldn't say this is a drug molecule, this isn't. And that is, in a nutshell, the reason it's so difficult. You don't know where to look for, which kind of structural class do you have to focus on. So the diversity is something that comes through. This is what I indicated I would mention. The ideal drug, and maybe this is for Simon to verify, but this is my view. We have something that's called the Lipinski rule of 5. You didn't mention that. Maybe you did most of it before those were published about 20 years ago. Lipinski was actually a colleague of Simon's, and he's become world famous. What did Lipinski do? I haven't met him myself, but what I'm told that he actually did is to look at the successful drugs, what's already on the market, and try to characterize them, to learn from them, and to avoid choosing the wrong compound. That's what it's all about. We heard about the thousands, maybe sometimes a couple of hundred would suffice, but you need to have criteria which ones to go for and which ones to leave aside. So that's the rule of 5. It's interesting. Molecular weight below 500. If you look at what this is now 20 years back, but still, we're not talking about 1,000 or 2,000 or 3,000 and approaching the biomolecules, small molecules. The risks that you approach when you go up in molecular weight is that you get metabolism, toxic intermediates in the metabolic chain of events. So keeping it small and not too many functional groups is per se good to avoid unwanted side effects. You have to look at the lipophilicity because we're talking about a water lipophilic environment in our bodies, and this is expressed as log P, this partition between water and octanol. It should be less than 5. You have the hydrogen bonds, which in themselves indicate how they can interact with receptors, and also not only donate but also accept hydrogen. So these are the rules, and the why 5? Well, 5 appears here, and this is 2 times 5. So it's a great rule. A lot of projects that I was involved with, that the discovery people worked on, they always check this. Doesn't mean you need to follow it to the nth degree, but it's a good guideline. That's the way I would describe it. But that's not enough. You need to measure somehow, and again, Simon went through this very elegantly yesterday. The efficacy, you need to have some way of assessing it. Is it selective, or does it hit many receptors? What about the bioavailability and ADME, as it's called in our jargon, absorption, distribution, metabolism, excretion properties? Because what you put in has to come out one way or another. We don't need to go into the details, but this is something which you need to have a look at pretty early on. We like to take our drugs orally. It's easier, you can self-medicate you as a patient. You don't need to go to a clinic to get an injection. So this reduces the burden in the health care system. Safety, obviously, it has to be safe in animal studies and later on in humans. It has to be stable. People often forget, I mean, hey, this is a chemical molecule. Not all chemical molecules are stable, are they? No. And here you need to have some criteria. Does it have to be kept in the freezer? I mean, these kind of things come to the fore. And running a business without some sort of protection is madness, because there are so many evil people out there who will steal from you, who will copy your compounds. We have many cases where others actually brought our new medicines to the market before we had launched them. So that's the way it's in the world these days. The public demands openness from the pharma industry. It has always been a very seclusive and a bit, well, isolated industry in that sense, not communicating too much just because you don't want to leak information that others might use for their own purposes. But being a public company, the shareholders are entitled to get good information. The market, as it's called, needs information about your projects. How are they going? And when you ask these kind of things, you cannot say, the project is okay. Don't ask. I call you, don't call me. Something like that. That doesn't work anymore. It worked when I started, but now you have to be much more open, you have to communicate, and therefore the patent protection that you can have on your product is extremely important. That's what you survive on. And I think, Simon, you can agree to that. Are there any medical needs out there? This is a bit far from the molecules, but I think it's important to understand what kind of diseases are we grappling with, and where is a major unmet medical need, which often goes back to poorly understand biological mechanisms. We know a lot about the human body, but there is so much more we don't know. So, stroke. It's the second or third most deadly disease in the Western world. It kills a lot of people, and those who are not killed by it, they get handicaps, speech disruption, hearing loss, movement of extremities. It's a very painful situation. You might survive a stroke, but it reduces your quality of life, that's for sure. Dementia. Not one day, at least not in Sweden, when dementia isn't mentioned in the media. And this comes with a population that is getting older and older. The risk of having a 50-year-old expressing Alzheimer's or any other dementia disease is very small, 1% maybe. But when you are 80 and upwards, it quickly raises to 10, 20%. And important to know is that Alzheimer's per se is deadly. The end point for Alzheimer's patient is death, nothing else. So it's not just that you lose your memory and you cannot communicate well with people. obesity, and it's an escalating issue, and you can see the numbers that are affected. 350 million. Type 1, that is the juvenile diabetes, and type 2 is the one that you acquire. Again, very many people are involved here. There are treatments. It's about glycemic control in the body, your carbohydrate metabolism. There is still much more to do. And the everywhere present cancer. It's a big area of diseases, and we can treat some cancer forms very well. Breast cancer is, the survival rate of in breast cancer is 90% or more. It used to be below 50 only 30 years ago. Prostate, the same. So there has been major inroads in the cancer treatment. Tropical diseases, TB, malaria, dengue, schistosomiasis, and what have you. This is a major threat, not for the whole world, but for certain parts of the world. And this is something that haunts people, and there are major casualties. TB, could it be 500,000 casualties every year? Malaria, somewhere in the same range. So people die more or less every second in these kind of diseases, and we cannot really treat them very well. We had a Nobel Prize in medicine a few years ago by this Chinese lady who was working with malaria therapy. This is a brilliant opportunity for people in Brazil, scientists in Brazil. You are living in the middle of it. In Sweden, we don't see, TB, maybe one or a couple of cases a year, but we are a bit bystanders. But in the countries where these diseases actually are in progress, I think you should take a lead and work with people from other countries, maybe with a more developed pharmaceutical industry, for example. The final disease area I want to mention is orphan diseases or rare diseases. Why is this a problem? The next slide will explain. This is data from a few years back, but the pattern is very similar. This is the definition. So in each and every orphan disease, we're not talking about millions of patients. We could talk about hundreds or maybe a few thousand worldwide. But this is frightening. At this stage, about 7,000 diseases were known that belong to this class. 7,000. Most of them, this high number, do not have any approved therapy. So all these people who are unfortunate enough to capture an orphan disease, you just sit there and wait for them to literally die, because most of these diseases are deadly as well. Genetic origin, they hit children because of you get your heritage and you're carried from the day you're born, and it can express itself right from the start. And this statistics is not very pleasant to see. So many children die before reaching the age of 5. It's a waste of human capital. So if people have challenged me, why are you developing drugs? We have so many, it's useless, etc. You just make money out of poor people or sick people rather. Yeah, to some extent it's correct, but there is so much more. It's like the top of an iceberg. So if there is someone maybe in this audience, who felt that making new medicines is not a worthwhile effort. I hope this can convince you. There is so much unmet medical need, both in the big types of diseases, stroke, cognition defects, etc., but also in this area of orphan diseases. Very bizarre diseases, and we don't know how to tackle them. This will be for your generation and for several generations to come to work on, if you are serious about creating good living conditions for people. This is another statistic about which areas do we see the failure in, and this is also a reflection on where do the pharma companies actually put their money, what are they investing in. Oncology, central nervous system, that's where all the Alzheimer and similar type of research is, and that's 50% at least. Musculoskeletal diseases, infectious diseases, then we are just looking at small pieces of the pie chart. So oncology and central nervous system, about 50% of the failure from a therapeutic point of view. Quickly, before I invite you to ask questions or comment, the role of chemistry. Where does the chemist appear in all this? We're here for the chemistry mostly, I think. The chemists are part of this, one way or another, as medicinal chemists and then as process chemists throughout the whole life cycle of the drug development. So again, if someone feels that chemistry is not important or doesn't contribute, show them this slide. Without chemistry, you would never have a drug. So it's a necessary evil in that sense, but it's not enough to produce a drug. But it's definitely something that supports it very strongly. From day one, when you work on the target, the biological target, making the molecules, the hits, the leads, optimize those, and then we take over and develop it into a process. This is what I just said. I was asked by Nature a couple of years ago to write an article, what is process R&D? Again, Nature is not per se a chemistry journal, but they do a few chemistry pieces now and then. I didn't have more than one page that was given to me, so I had to be very succinct and really home in on the main message. So that started with thinking, what should I call this piece of work? This was my first appearance in Nature. How should I frame what I'm doing? And then one afternoon, start small, think big. That's what we're doing as process chemists. Small, that's what discovery chemists do. A few grams maybe at most of a certain compound. That's where we start, but we have to think big. We need to think about the scenario. If this is a successful compound, we might be asked to do 100 tons. That is the size that comes in. So from day one, and I hope that my talk throughout this day will actually emphasize this very much, that you need to think ahead. Where am I going? What are my targets? What do I want to achieve? So chemical development is about synthesis of the molecules. It's about analyzing, keeping track of what happens in your reactions. This is not rocket science. This is what you all would expect. It's physical chemistry to really understand the kinetics. The reactions are complex. You need to be able to control them, to steer them. Their physical chemistry has gained importance. And chemical engineering. We work as a team. chemists and engineers. Only that way you can deliver the best process at the end of the day. So it's teamwork. We heard about the teamwork yesterday, and I can only emphasize this. Come up with the best route to the given candidate, and during the whole development stage, use material in the pilot plants mainly for clinical evaluation. That is the responsibility of a process R&D department. This is long before it becomes commercial, so this is only to support the R&D work. You need to evaluate which synthetic route fits the best, and from many aspects, including safety, environment, patent situation. You should try to avoid things that others have patented, obviously, because otherwise you would be in a situation where they might fine you, or you might have to pay royalty, and that's nothing you really would like to see happening. And the cost of the drug. It's often that you hear that the drugs are too expensive. People, at least in my country, they feel they're entitled to get everything for free. We have one of the world's highest taxes, and so people feel now the rest should be free. This has led to very specific and strong programs trying to drive down cost. And especially for underprivileged societies, they cannot afford, as it's called, the modern medicines. They are too expensive. They don't have the money. So what should we do? Should we give them away? I mean, this is an ethical discussion that we will not have here, but it's really something to think about. When you pay for a drug, you don't pay only for the active ingredient. That's a few percent. It's all this development work. We heard $1 billion. It could be $2 billion of dollars per registered new drug. And to be able to cover this, you need an input of money in your system. Otherwise, the machinery will come to complete standstill. No one will want to invest in this. AstraZeneca, when I left, had a R&D budget of about $6 billion. This means, what is it, 20 or so million dollars every day of the year. $20 million. And this is to do all this that is required by the regulators, etc., etc. Okay, and then at some point we will have to find which route do we go for. There are often many ways to go to Rome and also to go to a given molecular target. So which one do I go for? How do I pick the right one? This is the environment in AstraZeneca in Macclesfield, where I spent my last few years in the company, starting in lab scale. What would differ from what you are accustomed to is maybe a bit better control. Another type, these are cylindrical reactors. They are jacketed, so maybe a bit more sophisticated in that regard, to more look similar to the scale-up versions. This is a 50-liter glass vessel with a lot of tubings and valves and stuff. You don't see that very much, but start here, do your experiments, record your data, scale it up to maybe 50, maybe 100, maybe 250 liters, etc. And then eventually you land in the pilot plant, which is more like a factory. This should be very similar to what you see in commercial manufacturing. This is my first molecule. Day one, when I started in Astra, this is the molecule they offered me. Today, you would say it's a very simple molecule, and it is. It's not chiral. It has geometrical isomerism, but that's it. This was developed as a serotonin reuptake inhibitor, so it was an antidepressant that actually made it to the market. Can you believe that? The first molecule I worked on became a medicine. unfortunately it had to be taken from the market, because when you offer your product to a wider number of people, some not statistically significant side effects that you might know about from clinical trials, but due to the limited number of patients, you couldn't really rely on. But when you go out from 1,000 patients to 10 or 100,000, things start to happen. You see things. And here, and this is something that everyone who worked in the company then knew, it was the Guillain-Barré syndrome. Most of you, or maybe all of you, have never heard about the Guillain-Barré syndrome. It's an irreversible spasm in your face. It's a nerve contraction that is unfortunately irreversible. And it was a very rare side effect, less than 1% of the population. But in those days, Astra was too small. This was the first reuptake inhibitor on the market. We even came before, it was Billy, I think, who brought out the next one, follow on from this. We were too small as a company and didn't want to take the fight with the FDA. So we just backed off and took it from the market. In spite, so many patients had been helped, had taken them out of the deep depressions that you use these kind of drugs for. This is the synthesis. Convergent, that's nice. This arm and this arm, you come to this intermediate, and then you have a dehydration here. Yeah, looks good, but if you check what's said here, minus 70 degrees, a 3:1 geometrical isomer ratio. In the last stage, you lose a lot of material because you get this group sitting on the E side instead, which you cannot recycle or anything else. So this wasn't a really good route. I had to work like a slave to produce, what was it, first task, 500 grams of this material, just one day after the other. So that's when I decided I will go for a PhD, because when I started in Astra, I hadn't finished my PhD. So this made me realizing I need to be better at this. So this is the route I started with. So this said, how would you do this molecule in a different way? Think about it. What do you see a smart way of doing it that has less of cryogenic conditions? Minus 70 in the lab is easy, but in the factory, it's very resource intensive. It's expensive to run cold chemistry on large scale. And then to avoid this poor outcome in the dehydration, how would you make this molecule? Maybe you have read this paper. We didn't publish this until long after this project was already approved as a drug and then taken off from the market, but finally it was published. Any idea? Can you see how you can also in a smart, short way? The next slide will tell you, but I will keep you under suspense for a minute or so. Any throw out an idea? I mean, anything you see as a feasible approach? No, sorry? Wittig reaction? Yeah. Heck didn't exist. Well, he had published, but it wasn't in that common use. I think it was still too unknown, at least for us. You can probably find other things. No, it's fairly simple. This is what you do. So instead of bringing in the carbonyl function, here from this part, and the unsubstituted pyridine by pyridine lithium, you switch over the carbonyl to, this is nicotinic acid chloride, so this is a common building block with bromobenzene, and you run a Friedel-Crafts acylation. You get to this ketone, you can go down in temperature. This is a commercial Grignard reagent, adds very nicely. We are talking about 80+% yield if you trim these reactions. Then comes the beauty. You can see the outcome, 10:1. It's not entirely selective, but many times better than the previous chemistry. So what's happening here? It's an allylic rearrangement that you take. So phosphorus pentachloride, it's a nasty reagent, I agree. It is not very nice to work on, especially if you use hundreds of kilograms of it. But there are skilled companies out there who can handle these kind of chemicals. They have the safety precautions, so it's not a situation that resembles no-go. You can never do this. You can do it. You can work with diazomethane on large scale if you have the right technology. It's all technology driven. So what happens here is that the hydroxy group attacks the phosphorus here, kicks out the chloride, which attacks the double bond and relocates the double bond, and you get the allylic rearrangement, and then you just, and phosphorus oxychloride is the byproduct, and then you just substitute the chloride with dimethylamine, and there you are. Easy fix. So this is the process that we made commercial. Unfortunately, the drug died. This would have been a great drug by all means. That's it for the first part. Any questions? I promised you a breathing break, and if you have any questions or comments, now is the time. Oops. When do we have coffee break? We go on for half an hour or so? Okay, yeah, sorry. It's in the crystallization. Crystallization, I can hammer that message in, is the best unit operation for purification for pharmaceutical products. It's not always successful, but in 9 cases out of 10, if you can crystallize something and you find the right system, solvent system, this is a science in its own right, then you get almost entirely pure product. So it disappears in the mother liquor. That's the question, the answer to the question. The E isomer has a different solubility, so you can take it out by the crystallization. So the problem is gone. Yes?
What's the use of all the process?
The whole process, where did we end? When I started, I think I got, with this process, 0.5%. And the reason was that making this lithiation was not always under control. It's a matter of how you control your reactions. And if you have poor control, you don't know what to control, etc. So I think the overall yield was 3-4%. And that is not a commercial process, not for this kind of a short process. That was a no-go. So something else needed to be invented, and this was, yeah. Here the yield, I think, after the optimizations, 40%. The Friedel-Crafts reaction. If you can handle aluminium chloride, it's not the nicest reagent, but yes, 80%, Grignard's another 80% at least, and this well, you lose obviously material here, 40 to 50%, that's my guess.
So about the temperature, you told us that it's very difficult to achieve -70.
Yes,
So -40 is easier then because you can use a jacket reactor.
exactly.
And it works.
With -70, you need to work with liquid nitrogen. And that is very expensive technology. Here you can use normal brine systems and a heat exchanger, etc. So this is simpler at -30 to -40. It's expensive as well, but it's not so technically advanced as when you need to go to -70. Some companies still do it, and I've even seen processes where they go down to -100, but that adds to the cost, obviously. Okay, anything else? Yes? Yeah. No. Yeah, what are the solvents? Good question. Yeah.
Even on that scale, we have to get rid of the salt. And we don't have to pay for that. I mean, take the salt from the way out of it, yeah? So that's the first question. I noticed there was no salt in it.
Well, maybe my fault that I didn't include those. I thought more. This was interesting as it was my first project. Well, what do you have in Friedel-Crafts reactions? I know nitrobenzene is sometimes used so that you don't, you cannot use a solvent that could react under these relatively harsh conditions. I'll try to check it up. I don't remember, to be honest. This is, and I'm forgiven, this is 40 years ago, so people tend to forget me without having Alzheimer's. Sorry? Sorry?
40 years ago, you didn't do benzene or all the solvents which you might use today.
Yes, yeah,
Now, perhaps this 40 years ago, maybe all the time we had to use it.
yeah,
We have a little bit of solvent questions, even on that scale.
yeah. No, I know the situation has changed tremendously. And we in AstraZeneca, as have other companies, have produced a solvent guideline. The interesting thing is that the solvent doesn't know that it is a solvent. Only we can tell it, you are a solvent. I think that's very interesting. So we should treat the solvents with dignity. They are important players. They contribute to the chemistry. They're not there just as a bystander. They actively affect the chemistry to go the way you want to go, or you want it to go. I have to give this a pass. I need to either think it over or check it up. As I said, this is quite some time ago. Okay, I was given another half hour. Yeah, sure, sorry.
You somehow curious about how to deal, in your opinion, to treat orphan disease. Because orphan disease has a different status than how we deal, how we used to deal with neglected disease. And because you know that where metabolic disease conditions are quite even worse to their interest to put financial efforts
Investments, yeah.
to treat them. And what in your opinion, the future to deal with this kind of
I mean, the incentive for the pharma industry is that you get preferential treatment when you file for these drugs, and you often get a longer exclusivity on the market because there are so few patients. Unless you charge these patients outrageously much money, 10 times more than a normal treatment would cost, you will never recover the cost. It's difficult as it is with normal diseases, as we heard yesterday. I think only, 1 in 10, 1 in 5 or so drugs really bring in the money that you once invested in their development. And one of the reasons is obviously the price pressure, and the other one is all the generic competition and all the pirates that steal your inventions. And here the authorities are trying to offer you an incentive as a company to still, because the need is so enormous. But why should the shareholders of Pfizer or AstraZeneca, why should we pay for it? I include myself as a shareholder, if you excuse me. But anyhow, why should they pay for this alone? So it has to be, as we spoke about yesterday, a public-private partnership one way. The Health Organization, the United Nations, Bill Gates Foundations, the Wellcome Trust. I think it's only when we take away our pride and try to solve a problem rather than fight each other, that's when we will see an advancement here. Exactly how the model looks like, I couldn't tell you because I'm not that deeply involved anymore. But I think because so many smart people, or enough smart people, realize this. And I can say that because people talk about it. So many people are convinced that this is the way forward. So let's get it done. Let's join forces rather than going in different directions. Because the disease looks exactly the same in Sweden as it does in the US, as it does in Brazil. I mean, so why shouldn't we try to fight the disease together rather than in isolation? There's no need. Not in these rare disease situations. That's the best answer I can give you. Yeah? Okay. From molecules to processes. Half an hour more, is that okay? Still interesting? It is, yeah? Most people nod. Someone thinks the opposite? We are here again?
Yes.
Yeah? So what was the question? If we tried to... No. Not that I'm involved with. I did the part of the first chemistry, but then a co-worker of mine took over, and I don't think, once we identified this route, Wittig is in the lab relatively easy, but it's also a tricky reaction to control on the large scale, so we weren't really comfortable. So we felt that Friedel Crafts is, after all, our best choice. Maybe they did the experiment, but the yield didn't come out well, and then they just put it aside. Yeah, it's a good idea, definitely. Okay, from molecules to processes. Now I will tell you about a very spectacular case that I definitely was involved in. And I could have said from the beginning, when I started my career in Astra in the 1970s, it wasn't customary for process R&D people to publish. People in discovery published. They showed the molecule, they gave information about the physical properties, the screening, the affinity for receptors, etc. That was commonplace. But to go out and talk about how you develop what might become a commercial process, that was a bit too dangerous, because you would reveal, first thing, which projects might actually end up in becoming a medicine. Because what discovery does, there is still a very high attrition, if you remember this, one of the first slides. A lot of discovery projects will never go anywhere. So you can always disguise which molecule is the most important one. In development, people will know that we only work with the molecules that have reached a candidate drug state, meaning there is something here. So this could lead people in the direction that we don't want them to go. They could start thinking about and starting to make analogs and stuff like that, check our patents to see if there is a patent opportunity for them, etc., etc. So it wasn't customary to publish. I was one of the people who realized if I want to have a foothold in academia, I need to publish. So I started this on small scale in the beginning, but this has never been a normal part of our work. We don't need to publish, but if you want to, well, be invited to these kind of events, who would know about me unless I published? I mean, that's the name of the game. You don't read our files, so we need to publish in the open literature. I think this has been, and this is now seen by Pfizer, by Merck especially, who pioneered publishing from their process research and development. They were once, and I think still, one of the top-notch companies in process R&D. I know them very well, and this is a situation where we have not only published on a process that we scaled up, but actually on a process that became a commercial success. Omeprazole, you saw the molecule, the sulfoxide earlier on. This is the story about it. And I published this in several fora. The biggest is probably this book chapter that I wrote with my co-worker a couple of years ago. The first time this molecule was made was in 1979. Sulfoxides are chiral molecules. If you have two different R groups on each side of the sulfoxide moiety, they are chiral. They are they have 4 bonds, even if one is just a lone pair electron. So they have chirality in them. And this was realized when omeprazole was launched as Losec, pre-Losec in the US by Merck. We were in collaboration at that time. But there was no technology available to separate them. And all the tox studies were done with the racemate and worked well. It was a safe drug. This is a relatively short-term treatment of 2 weeks or something to treat ulcers, stomach, gastroesophageal reflux disease, etc. All that has to do with the acid production in your stomach. In the meantime, during the development of omeprazole, these 10 years, there were attempts to separate the enantiomers, to document both of them, both the R and the S form, but only in very small and very specific columns that analytical specialists worked on. So this was nothing that would ever go commercial anyway, if the technology wouldn't have improved. Again, a bit larger quantities, we talk about a few milligrams, just to run a tox study or something. Losec was launched and became a world hit, and about 10 years later, here in the mid-90s, it was the world's single largest selling drug, $6 billion at the time. So as a company, this was still Astra. was doing very well, obviously. But once larger quantities of both enantiomers, the S and the R form, had become available during these years when the technology developed a bit better, because you couldn't make them chemically. You had to make the racemate and then separate. There was no chemical means that you could make one of the enantiomers in good enough quality and yield. Then this became the most prioritized project, because in the pharmacokinetic studies there was a very clear effect advantage for one of the enantiomers, quite unexpected. And when it's unexpected and when it's novel, you can patent. And that's what we could do. Otherwise, nowadays, to go from a racemate to pure enantiomers, that is a no-brainer. You can do it. There is technology. So it's exceedingly difficult to get a patent protection if you go from a racemate to a pure enantiomer. Now, in those days, it was still possible. So we started a project that was not allowed to fail. And here, this is probably the project I felt the strongest pressure from the upper management from. We were constantly bombarded with questions, how does it go? Because here we were on the critical timeline. Normally chemistry is never on the critical timeline, unless it's in the early stage when you have to prepare a first pilot batch and you don't have control of your chemistry. But otherwise, it's more the tox studies and the clinical studies later on. That's the critical timeline. But here it was chemistry, because without having access to the chosen enantiomer, this project would have stood still. And if we would have said, God forbid, we can't make it, we have to give up, then most of us would have been fired, I would assume. We weren't, because we were successful. So after huge efforts, we managed to get an asymmetric process in place, which we then developed. I will show you how it looks like. And it was scaled up in record time, and it was transferred to commercial manufacturing, batches of more than 100 kilogram. And it was launched as Nexium. The active ingredient is now not omeprazole, but it's esomeprazole, and you will all figure out why it's es. It is the S enantiomer. Smart way of hiding the absolute stereochemistry. So the S enantiomer was launched 2000 and became a hit. It was brilliant. We did some extra efforts trying to even improve further on this, but we never succeeded to bring out a third compound building on this omeprazole structure. So, we're talking about proton pump inhibitors. The original project was actually started more than 50 years ago in Gothenburg at a small part of the then Astra research organization. And they had focused on gastrointestinal diseases, ulcers, reflux disease, very painful. This is a brilliant paper of colleagues of mine in Discovery, who also in Nature have described the early stages, how they came up with this molecule. How did you treat ulcers in those days? Antacids, you neutralize the acid. You need the acid, but you don't need the acid in the wrong place and too much of it, because then you start to have corrosion in your stomach, in your linings. And that's what creates inflammation and very painful stomach problems. A lot of surgery was used, gastrectomy, vagotomy. You just cut out pieces of your stomach. It doesn't sound so tasteful, but that's how it was done. The surgery by itself wasn't very lengthy. You open the stomach, it's open surgery in those days, obviously. but the patient had a very long convalescence afterwards, a month, 2 months, and there was no guarantee that it wouldn't come back again, so you would have to go through another surgery, etc. Novel medicine started to appear on the scene. Tagamet, this was SmithKline French, who came out with a drug that really worked beautifully to reduce the acid production by working on the histamine receptor that was identified inside the stomach, inside the lumen, and then even more ranitidine, the Glaxo star, which really took them to novel heights. They, extremely successful drug by all means. They were introduced in the late 1970s, and it antagonized the histamine 2 receptor. Other compounds were identified that had similar properties. Interestingly, when this early work was done, no one knew about this enzyme because it was published only in the late 1970s by an American team that had identified this hydrogen potassium ATPase as the enzyme that is responsible for the ultimate release of a proton. This is brilliant. Again, it shows how important enzymes are. That's the reason I love working with enzymes, because they are mysterious. They can do all sorts. They are good tools for making chemistry. They are involved in a lot of physiological transformations, etc. So once this became established, once you knew what your target would be, this is a secondary target, meaning that the treatment control isn't as exquisite as it is when you work directly on the enzyme that gives the proton. That's what we did. First compounds were tested in man. They were inefficient, and this is again the dichotomy between working in the right animal model and draw conclusions from those studies for their efficacy in man. Sometimes this works good. This is a common practice. You work with an established animal model and then try to interpret this. How will it work in man? Here it didn't. It worked in the rat, people were happy, but not in man. So, dead end. But a dog model. We had some really smart toxicologists in the company. So one of the first things when I had started in Astra, I went to visit this company in Gothenburg, this part of our company in Gothenburg, and they used the beagle dogs. For this is a common breed of dogs that you use because they have the right size. Labradors are too big, others are too small, Chihuahuas, but beagles with a body weight of about 10, 12 kilograms, perfectly suited. So they had developed this dog model. They were catheterized, so they could measure all sorts of data whilst the dog was standing in a cradle, in a sense. And in the beginning of 1979, so it's 40 years ago, omeprazole was first synthesized. This is the synthesis. After a while, I have to say, after we had worked on it for a while, this is not the way that it was done initially, because this as a starting material is far from trivial, so we had to invest quite a lot of time making this pyridine alcohol. But otherwise, from here on, it's a child's game. Chlorination, quantitative, you bring in this benzimidazole, again quantitative. You get the sulfide, the prochiral sulfide, and then you oxidize it, standard procedure, beta-chloroprobenzoic acid, toluene. Here you have solvents. Tim, I haven't forgotten them here. You get omeprazole, the racemate. Perfect. Straightforward. The first time we were asked to make the enantiomers, we were asked to make both because no one knew which one was better than the other. So we had access to omeprazole, obviously, as much as we wanted. So for us, it just became a matter of separating the two enantiomers because there was no asymmetric synthesis available at the time. Obviously, everyone realized that if we can find an asymmetric oxidation, we'll be home free, but it took some time. Slightly busy schedule, but it was a busy way of separating these enantiomers because you cannot crystallize them apart. Otherwise, people would say, this is a piece of cake. No, it has to be, and I won't go into the details. You can read the book chapter, it says it all. But we had to come up with a chromatographic separation. And in order to do that, this is the racemate, you need to derivatize omeprazole with this hydroxymethyl group. And here, you get both the 5 and the 6 in isomer because both nitrogens in benzimidazole are active. Both will react, and you can use both. So that's a smart way. So you don't need to separate anything here. What we eventually wanted to do is to separate a diastereomer mixture. That's the reason we went through this. Because again, at this time, there was no chiral stationary phase available at the quantities we needed. Otherwise, you could run it through ignoring the cost of these stationary phases. You could do it. But for making kilogram quantities, we had to circumvent the use of chiral stationary phases by this derivatization, the hydroxymethyl to the chloro, and then attach mandelic acid, one enantiomer. It's a very lengthy and tedious process to prepare the molecule for chiral chromatography. This is a simple chromatography because we talk about diastereomers. What was not so simple, what we found out, is that once you have separated the R and the S, these are not stable in solution. It's a very tricky molecule, omeprazole, and it mutarotates. So unless you work very fast, the pure enantiomer will undergo mutarotation, and you end up in a racemate. So people literally had to work 24/7 to make this happen. It was a tour de force to do this. It was a fantastic team effort, but we succeeded. We prepared both, about 0.5 kilogram, what became isomeprazole and the R, and the pharmacologists had enough material to start documenting it in much more detail. Oops. There we go. But a sunny day, we came up with what I'm showing you now. One of my best achievements in process chemistry. We found a way to go from the prochiral sulfide to the desired enantiomer. We tested, this is about 1991, maybe, two. We tested the best available technology to make chiral sulfoxides. This is the Kagan chemistry, building on what Sharpless also did, but not on sulfoxide, on sulfides, but on alkenes, for example. So you talk about titanium, you have diethyl tartrate as a chiral element, and you use an oxidant. And if you have simple sulfides, let's say phenyl or benzyl here and methyl or ethyl or something, you can achieve a 90+% EE sometimes in one go. Because when we tried the best known technology from the Kagan methodology, we got 5% EE. 5, not 50, 5. So what this tells you is that, yes, it works. It's at least a glimpse of hope, but you need to refine it. And that's what we did. And the clue to this is this beast. Unique space. When one day because the molecule is acid sensitive, and still it inhibits the production of acid. It's quite a funny situation. It's acid sensitive. So by having a base present, we thought maybe we can protect, we give it a better environment, etc. We really didn't know what we were talking about, but this was a chance. And we managed to get, after some polishing, etc., more than 90% EE. And this is still quite unprecedented, quite fabulous to have this done. So this is essential. We didn't understand what the Unix base actually did, how it operated from a mechanistic point of view until later, after the launch. But when you launch something, when you register a new medicine, you don't need to demonstrate the mechanism of how you make the molecule. As long as you can prove that the quality is impeccable, it's standard, you have the control of the process, the regulators will be happy with that. A mechanism you can never patent anyway. It's like a mathematical theory. You cannot patent mathematical theory. The same is with a mechanism, even if it's a lot of work behind it. So what we had achieved here, finding this base, we understood that you need to pre-formulate the catalytic species, and we do that in step 1. We totally changed the solvent system, the temperature regime. Normally these reactions, at least in the hands of Kagan and others, are used dichloromethane, minus 20-ish degrees, something like that. We could go in the opposite direction. So much more process friendly in that sense. An oxidant, very cheap, cumene hydroperoxide, much cheaper than the racemic process where metachloroperbenzoic acid is. The oxygen equivalent you use in metachloroperbenzoic acid is extremely expensive compared to cumene hydroperoxide. Again, very decent conditions. We have shown, and our patent says, between 4 and 50% titanium. This operates very nicely. The turnover numbers, meaning that every catalytic species can convert 4 to 16 molecules of the substrate, and the turnover frequency, that's just over time. So the reaction is over in a bit more than an hour. You wouldn't be too impressed, for those of you who work in catalysis, with turnover numbers, 1 or 2 digit numbers. You have to remember this is an oxidation, and oxidation reactions are much more demanding from a chemistry point of view for the catalyst than reductions. In reductions you see 100,000, etc. reported. That's commonplace. If you are operating with 10 or 20 in a reduction, you won't even be allowed to publish in a journal of the obscure type. So. But this is pretty good, and there are not so many asymmetric oxidations out there. When we did this work, and when this book was published, and also the book that Blaser and I published some years later, only about 15 to 20 asymmetric commercially running processes were known. So whilst academia is investing a lot in asymmetric chemistry, industry doesn't use it. something food for thought. It's an interesting observation. Don't know what experience Simon has, but it's striking. And the reason for me is quite obvious. It's because so many projects where you have asymmetric steps fail. Not because they are asymmetric, but because so many projects in the pharma industry fail. That's the driver. So all these fantastic asymmetric processes that have been developed, they are, well, some have been published, but others are buried in people's cupboards, unfortunately. So on the surface of it, it seems that asymmetric technology isn't so important. You couldn't be more wrong. It is. We couldn't live without it. So we have produced several hundred tons of this material, always with the same performance, 90% yield in this step and 90% EE. It's so robust. It's fantastic. I get all excited, so excuse me. I needed to show also a flow chart of this process, because when you are a process engineer, you don't like to see this kind of stuff. It doesn't mean so much. You want to see what are we actually doing in the factory, and there the process flow charts come into play. So this is the same chemistry as you see here, but expressed in a different way. This more homes in on the unit operations. What are you doing? You are charging, you are stirring, you are separating, you are working up, you are isolating. That's what these things are there to illustrate. And before the break now, only several years later, we managed to come up with this catalytic cycle. I said, I will stay active as a chemist until the day when I know the mechanism for this self-oxidation. And this is what we published. It's a group at the Stockholm University and myself. We had an Indian postdoc with us who did most of the groundwork. In this pretty comprehensive paper, we also had to use quantum mechanics to do some calculations. We could never isolate, even if we tried, the catalytic species. But I think we have good indications, indirect, albeit, for what it is. The interesting thing is, for those of you who know your Kagan chemistry, you will know that you need to have some water. And I haven't seen any explanation why you need this water. Why do you have to add the water? Now we know. Titanium complexes of this sort are dimers, or even oligomers. So what the water does, and we have shown this quite significantly, is that it de-dimerizes it. And this is the active titanium species. And then you have some ligand exchange here, and you bring in the oxidant. You have a periplanar arrangement. As you can see, it's nicely depicted here, and you will see this when you get the slides. This, we used imidazole as a model because that's much more easy to work with from an analytical perspective than the substrate that we had in the omeprazole case. And so this alignment is very strong between the oxygen that is attached to titanium and the sulfide, and it's phase selective. It explains why you just get the attack from one phase of the molecule and not the other. And this brings you this beautiful outcome. I'm still very proud of this. Yeah? I'll end here for a break. Are there any immediate questions, or you need to digest what you just heard? There is one question.
The oscillation of the circle
Yes, of course, it's patented. But yes, you could go either way, either from the sulfide to the sulfoxide or from the sulfone and reduce. It's been covered. But in our hands, and this is now 20 years ago, our skills in biocatalysis wasn't so developed, if I express it mildly, as it would have been today. So maybe with the current knowledge and access to much more refined enzyme systems, that might have come out as the process of choice. So yes, we did look at this, but the chemical way was so outstanding, as you can see from the performance data. Cheap titanium costs nothing, the oxidant is more or less for free, so everyone was happy, and we managed to get it to the market on time. So yeah, that's the response, but you're absolutely right, it's possible, we know that. Yeah?
Thinking about nowadays, like if you were preparing some chiral molecules, when would we use chiral chromatography and when would we use selective synthesis, like which scale and which timeline in the process, when we would choose one or the other?
The direct and a bit aggressive answer is never. We would never use chiral chromatography. The people in discovery, that's a different ball game. They are only, their task is primarily to produce new molecules and to have them tested. In development, you need to think, as I had as my headline, start small, think big. You could never envisage, at least not from where I'm coming, to run chromatography on a scale of 100 tons. So yes, it's a last resort. We wouldn't go to the management and say, we can't make this because we don't want to work with chromatography. Then the response would be, there's the door. Don't come into the door again. You have to use the technology that is available, even if it costs a lot, knowing that in the beginning the volumes are small, and the likelihood that the project will succeed is also very small. So maybe you just will do this once, and then you will see that this is a dead end, the molecule doesn't work. You can shelve it. So yes, if nothing else works, we will use chiral chromatography. It's case by case. It's maybe not the answer you want to hear, but it's always case by case. If the case is the right, the technology is available, let's do it. If the case is wrong, technology is not there, you have to do something else. Is that
when you're just investigating the molecule, let's say not speeding up the process, then it will be the time to use.
Yes, yeah, sure. Chromatography as a unit operation has really come to the fore as a good tool. You can operate this. We had a preparative chromatography unit, and people threw all things at the columns there and separated whatever stuff you have. It's a good purification method. The problem is to go to the larger scale. So, but it can be done, and some companies have pioneered it and are also producing commercially, where chromatography is one element in the process. So yes, you cannot be too stubborn and say, because we've never done chromatography, we will never do it. Then you are in a dead end. You need to be open-minded and take the best technology. Okay, let's stop here.
I suppose that